In Jurassic Park, the promise was clear: we spared no expense. The 1993 film made cloning dinosaurs from ancient DNA seem just a heartbeat away. But the real reasons we cannot bring back dinosaurs are buried millions of years deeper than Hollywood lets on. DNA's half-life is only 521 years. After 6.8 million, every genetic code is gone, yet dinosaurs disappeared 66 million years ago. If the science is this conclusive, why won't the fantasy die? The truth upends everything you think you know about extinction and resurrection, so what really stands in the way? In the summer of 1993, moviegoers watched in awe as a mosquito, trapped in golden amber, held the key to a lost world. Jurassic Park didn't just entertain, it rewrote what people believed was possible. The idea of extracting dinosaur DNA from preserved insects was more than just a Hollywood invention. It drew inspiration from a 1992 Science article where researchers speculated that ancient DNA might survive for millions of years. That spark fueled a wave of headlines and scientific curiosity. Suddenly, the line between fiction and reality felt thinner than ever. Museums displayed fossilized amber, and news outlets ran stories about genetic resurrection. The public imagination latched onto the hope that, with enough ambition and technology, the distant past could be brought roaring back to life. But as excitement grew, scientists began to question whether the evidence behind these claims could withstand closer scrutiny. The search for real dinosaur DNA would soon face its first major test. Fossils may look like time capsules, but their secrets are locked in stone. When an animal dies, its bones don't simply wait to be discovered. They undergo a dramatic transformation. Over thousands, sometimes millions of years, groundwater seeps through the remains, carrying minerals that gradually replace the original bone. This process, called mineralization, turns living tissue into rock. Any cells and the DNA they once held are erased by chemistry and time. In 2005, paleontologist Mary Schweitzer made headlines with a Tyrannosaurus rex femur that contained soft, flexible structures. Some even called it a breakthrough. But what Schweitzer actually found were fragments of collagen, a tough protein, not DNA. Collagen can survive under rare conditions, but it cannot store genetic instructions. Despite decades of searching, no one has ever recovered dinosaur DNA from a fossil. The bones that fill museum halls are silent stone records, not blueprints for resurrection. The hope for ancient genomes fades with every mineral crystal that replaces living matter. DNA is not built to last. In 2012, researchers studying ancient Moa bones calculated that DNA's half-life, the time it takes for half of the bonds in a strand to break, is just 521 years at average burial temperatures. That means after only a few thousand years, even the largest fragments begin to splinter into unreadable pieces. The math is relentless. By 6.8 million years, every chemical bond in a DNA chain has snapped. Not a single readable sequence remains. Even in the coldest, most stable environments on Earth, the oldest confirmed genome comes from a horse bone, frozen for about 700,000 years. Far short of the 66 million years since dinosaurs vanished. Temperature, moisture, and even the soil itself speed up decay, making survival of genetic material impossible over deep time. The numbers leave no room for hope. The very chemistry of DNA draws a hard line that no dinosaur genome can cross. Cloning is not a matter of wishful thinking. It depends on biology's strict rules. When scientists cloned Dolly the sheep in 1996, they started with a living cell. Inside that cell, the nucleus held a complete set of DNA instructions, preserved and ready to be reprogrammed. The process, called somatic cell nuclear transfer, required a functional nucleus to guide the development of a new organism. Every successful clone, from Dolly to more recent animals, has relied on this fundamental ingredient, an intact genome inside a living or recently preserved cell. In the case of dinosaurs, this basic requirement is missing. Fossilized bones, mineralized over millions of years, contain no living cells and no nuclei. The original DNA, once housed safely in those ancient cells, has long since vanished. Without a preserved nucleus, there is nothing for cloning technology to work with. No blueprint, no instructions, no possibility of bringing a dinosaur back from extinction. The door to cloning for creatures lost to deep time remains firmly closed. Piecing together a dinosaur genome would demand an almost complete set of genetic instructions, over 90% by scientific standards. With dinosaurs, nearly all of that information is missing. Researchers estimate that about 99% of the original DNA sequences have been lost to time, leaving only the faintest traces, if any at all. Modern gene editing tools like CRISPR-Cas9 have revolutionized medicine and agriculture, but they work by tweaking known genes, not by inventing or guessing massive stretches of lost code. Imagine trying to rewrite a novel with only a handful of random letters left. No amount of editing can fill in the gaps when entire chapters are gone. Even the most advanced sequencing technologies can't reconstruct what isn't there. Without a living relative close enough to provide a genetic map, the gaps in a dinosaur's genome are simply too wide to bridge. The dream of rebuilding a dinosaur from fragments runs into a wall that no tool, however powerful, can break through. Jack Horner, a paleontologist with a lifelong fascination for dinosaurs, set out to answer a question that captured the imagination of scientists and moviegoers alike. If we can't recover dinosaur DNA, could we reawaken the ancient traits hidden inside their living descendants? In the early 2010s, Horner's team began experimenting with chicken embryos, searching for dormant genes that once shaped dinosaur features. By tweaking the genetic instructions, researchers coaxed chicken embryos to grow teeth, sprout elongated tails, and even develop snout-like faces, traits lost millions of years ago. These dramatic changes offered a glimpse into the evolutionary past, confirming that birds carry a genetic memory of their dinosaur ancestors. Yet, even with these breakthroughs, the results remain firmly anchored in the present. The so-called Chickenosaurus is, at its core, still a chicken, its DNA shaped by countless generations of avian evolution. The experiments reveal the deep links between birds and dinosaurs, but they also draw a clear boundary. Modifying a chicken's genes can highlight its ancient heritage, but it cannot bring back a true dinosaur. The genetic gulf is simply too wide to cross. Even if scientists could somehow piece together a dinosaur genome, the next roadblock is biology itself. Cloning a mammoth is possible because elephants and mammoths share about 99% of their DNA, making an elephant a suitable surrogate. Dinosaurs have no such counterpart. Birds are their closest living relatives, but more than 65 million years of evolution separate them. A dinosaur embryo would need the right environment to develop, but a bird's egg is the wrong size, shape, and chemistry. The proteins in the shell, the nutrients inside, and even the temperature needed for incubation are all mismatched. No living animal can offer what a dinosaur embryo would require to grow. Dinosaurs thrived in a world nothing like our own. During the late Cretaceous, oxygen filled about 26% of the atmosphere, 5% higher than today. Carbon dioxide levels reached 5 to 10 times current concentrations, fueling lush vegetation and a warmer, denser air. These ancient reptiles depended on conditions that no longer exist. Modern ecosystems are shaped by different plants, animals, and even microbes. Introducing a dinosaur would mean forcing it into an environment it was never built to survive. The risks go beyond the animal itself. Such a disruption could ripple through food webs, threaten existing species, and create unforeseen ecological costs. Birds carry the living legacy of dinosaurs. The fossil Archaeopteryx, with its blend of feathers and reptilian features, bridges the gap between ancient theropods and modern flocks. Genomic studies reveal that today's birds share deep DNA ties with species like Velociraptor and Tyrannosaurus rex. The evidence is clear. DNA's half-life of 521 years, as shown in the 2012 proceedings of the Royal Society B study, sets an absolute barrier. After 6.8 million years, no readable genetic material remains. Dinosaurs vanished 66 million years ago, making recovery of their genomes impossible. No dinosaur fossil, including Mary Schweitzer's 2005 T-Rex find, has yielded DNA, only protein fragments. Attempts to reverse engineer traits in birds like Jack Horner's Chickenosaurus experiments confirm evolutionary links but do not resurrect extinct species. Key gaps remain, such as whether future techniques could recover more information from trace proteins, but The core obstacle, absence of intact DNA, remains unchanged. Today, birds are recognized as living descendants of theropod dinosaurs, supported by fossil and genomic evidence. The search for dinosaur DNA has reshaped molecular paleontology and underlines a simple fact. While we cannot bring dinosaurs back, their evolutionary legacy continues in every bird alive now.